Very happy to have Jeff Flatten here today with us from the Foldit team. I first heard about this a little more than a year ago, and then got an opportunity to hear about the project about a year ago at the NIH. And I'm not going to attempt to tell you the project, I'll let Jeff do that. But Jeff's a developer working in the Center for Game Science that's located in the Paul G. Allen Center for Computer Science and Engineering at the University of Washington in Seattle. And he graduated from the University of Washington with a BS in computer science in 2010. He's been part of the Foldit project since October 2010. I'll just briefly say it's an online game, multiplayer video game that enables anyone to contribute to real scientific discoveries in biochemistry. And the project I heard about was one of the protein folding problems that were solved using game theory. And rather than try to interpret that any further, I'm going to turn it over to Jeff and let him tell you about the project. Thanks for coming.
Thank you, Dr. Davis. So what I'm going to be talking about today is Foldit and games for scientific discovery. So the idea is that games are playing an increasing, more important role in today's society, and it's really a growing field. And in a lot of senses, a lot of the effort that people are pouring into these games is being wasted. So the game developers are obviously making a living off of this, and people are getting enjoyment out of games. But what if we could actually harness all of this energy that people are pouring into these games and actually use that and direct that towards some sort of goal? So in particular, what we're interested in doing with the Foldit project and with projects like it is actually setting things up such that the effort that people are putting into the game is going to go towards scientific discoveries. And so in Foldit, what we've actually done is made a protein folding video game, where by playing the game, players are going to help solve protein structures. And these protein structures will help better our understanding of science of the human body and just scientific goals in general. So proteins are sort of, you can think of them as small machines within your body. They do all the basic work within the cells. Some functions of them include protecting from diseases, transporting other molecules, and manipulating DNA. So a key thing about proteins is that their three-dimensional structure, what shape they take, is actually what determines what their function is. So here you can actually see a lot of different proteins. These are all real proteins, and they come in a lot of shapes and sizes. And they all do very, very different things. So at its most basic level, a protein is actually just a sequence of amino acids. So when I say a sequence, what I actually mean is a protein structurally is like a chain, like a string of pearls. And each pearl on this chain is an amino acid, which is a smaller molecule. And they're strung together in this string of pearls, and that's what we call the sequence. And you can see a sequence here, the MQIF. Each of those letters represents an amino acid. And these are strung together in that order, like a big long chain. And a protein, every sequence, every protein also has a three-dimensional structure associated with it. And every sequence will actually fold up into the same three-dimensional structure every time. So each sequence has its own unique three-dimensional structure and its own function as a result of that. So in general, it's pretty easy for us to determine the sequence of a protein, but much harder for us to determine the three-dimensional structure of a protein. And again, that's actually really useful for us to know the three-dimensional structure, because that tells us what the protein does. So there have been a number of different approaches towards solving for the three-dimensional structure of a protein. The first attempts are, well, you would think, obviously, just look at them and experimentally try and figure out what these look like. And they're actually so small that you can't see them with microscopes. So we have to use different techniques. And these techniques are slow and expensive and do not always work. So sort of another approach that has been taken is computational methods. So what we do is we run a simulator on or just a predictor algorithm that takes the protein sequence and tries to predict the three-dimensional structure from that sequence. And here are two such projects. There's Rosetta at Home and Folding at Home. Both of these are set up as screensavers that people can run on their home PCs. And whenever they're not using their computer, this screensaver will run and donate some of their idle processing time towards trying to solve protein structures. Now, these projects have been met with reasonable success. They've solved a lot of structures. But in general, protein folding is actually really hard, and just trying to do it computationally doesn't really work. There's just too many degrees of freedom within a protein, and we can't just tell a computer to crack away at it and have it try and solve it, because it doesn't really know how to search the space very efficiently. So why would we turn this into a game? The first thing is exactly what I've just said, which is it's computationally difficult. And computers alone are having trouble trying to search this space for the correct fold of a protein. And the second is that humans actually possess very good three-dimensional spatial reasoning skills, and also very good problem-solving skills. And proteins themselves are actually very puzzle-like to begin with. They're three-dimensional, and there is some desired shape that we're trying to find. And through a series of manipulations, you can find the correct structure for that protein. And so it's a very natural problem-solving space. And lastly, another good contributing factor to why this makes it a good candidate for a game is the impact that it might have. So people play the game because they are trying to contribute something, and whether it be deepening our understanding of how the human body works, genetics, or trying to combat diseases. Okay, so the video did play. So here's actually the game Foldit. Somebody is playing it right here. You can see the person is sort of dragging the protein around, manipulating the three-dimensional shape of it. And as they're doing that, you can see their score up near the top is changing depending on what they're doing. So Foldit has been pretty successful so far. We have over 100,000 players. We have over 100,000 players, and we've had several publications, including in the journal Nature. So the way that we've set up Foldit is we take open problems, or scientists will come to us with open problems. And we take the open problems are we're trying to determine a particular structure. We'll post that to our servers as a puzzle that players can then download to their game client. And these puzzles are like actively posted. We post a couple a week usually, and they're open for about a week at a time. And while they're up, people have the opportunity to work on them and try and find the right structure. And as they're doing that, they're sending solutions back to our server. And we can get those solutions off of the server, and we do some analysis on that. And based on the results, we can either sort of send them off to the biologists in the lab to have them look at them if we think that they're correct, or we can send it back as another puzzle and tweak some parameters to try and increase the results or get better results. And this cyclical aspect of Foldit, of sort of iterating on the design and tweaking things and sending them back out, is very core to the design philosophy of Foldit. And it applies not only in just puzzles, but also in game updates. So we do a lot of work to increase the features within Foldit to be able to tackle new types of problems. And in return, we also get a lot of player data back. This is data other than the actual structures that they're generating that is also really important to us, such as which moves they're making when, how they're manipulating the protein, how they're viewing the protein, because all of this is extremely important as well. So this is that scoreboard that was up at the top of the video earlier. It turns out this score function that you see here, within biochemistry, the scientists don't talk about score, they talk about energy. So the energy of a protein is exactly that. If you have a lot of energy, the protein is somewhat unstable. And in nature, a protein tends to try and find the lowest energy state. We call this the native of the protein. So the protein will sort of move around and shift and change conformation until it ends up in the lowest energy state. So lower energy is better. So one of the really big design philosophies behind Foldit is we want to make it as accessible as possible and keep it within the game's framework. So one thing that we did was we took the score, or the energy, and added, I think it's added 800 and multiplied by -10 in order to make this a large positive number, because people like that and people are used to that in games. And so in addition to your score, you'll also see your rank. So Foldit is set up as a competitive online multiplayer game. So you're folding proteins. You're also seeing how well other people are doing on folding those proteins. And so here you can see we have group competitions and soloist competitions. So soloist is just you working on that protein. And for the group, it allows you to collaborate and work on the same protein and try and come up with better results through that. So another area where we've tried to make this very game-like is the visualizations of the game. So with the visualizations, our goals were to make it simple and have them clearly demonstrate the basic rules of protein folding, because it turns out you can actually reduce protein folding to some fairly easy and able to be understood principles. So this right here is not a screenshot of Foldit. This is actually a screenshot from a piece of biochemistry software called Pymol. And it probably looks somewhat confusing. It looks like a bunch of green sticks connected all, and I can't really tell what's going on there either. So in the game, we tried to just highlight the relevant information and really keep it simple and clear. So I believe this is actually the same protein in both of these screenshots. So with ours, you remember I was talking about a protein being sort of a string of pearls. And the string, biologists call the part that connects all of these different amino acids, that is called the backbone. So you can actually see in the Foldit's visualization, the backbone runs across the entire protein, and you can sort of trace where it goes through. It might be kind of hard to do that in the other one. And in addition, the other big part of proteins, there's the backbone, there are also the side chains, and each amino acid will have this little bit that's hanging off that we call a side chain. And in the Foldit screenshot, you can see the side chains are the blue and orange pieces that are hanging off. And they're actually partially hidden because it's not totally important for people to be able to see those all the time. And another big area where we've tried to really keep the information as useful as possible is the score. So with the scoreboard, you can see your score at any given point, but that doesn't really tell you too much about what areas need improvement or what areas are doing well. So what we've done with our protein in this visualization is we actually color the protein and the subregions within the protein based on how well they're scoring. So the green regions are scoring better and the red regions are scoring worse. And so that gives you an idea of, hey, maybe those red regions, I should try and work on those. Those aren't in the right shape. So the basic rules can be demonstrated by some pretty basic geometry and visualizations. And this is all in the theme of keeping things as clear as possible and accessible to people who are not biochemists. So one area where this happens is with clashes. So when two atoms are too close together, they tend to try and push apart with a lot of force. And so this leads to a very high energy protein. And in general, you don't want that. So when this happens, you'll see this little geometry, this red spiky ball, when two atoms are too close together. And that'll result in a very bad score, but it also tells you where this is happening. Another area is with voids. So in general, proteins want to be as small as possible, as compact as possible. And if you have a large region of unfilled space inside of your protein, it's somewhat unlikely because the protein would have reconfigured itself to try and fill in that space. And so that's what these red balls, spheres, represent, is a place of empty space within the protein. And hydrophobics are another area. So a hydrophobic is a type of side chain within proteins. So there's two types of side chains within proteins. There's hydrophobics and hydrophilics. So hydrophobics are afraid of water. That's what hydrophobic means. So they will try and hide from water. And proteins are floating in water. Sort of like just, if you were to dunk the protein, it's sort of like this ball, and you dunk it underwater. The place that is most hidden from the water is in the center of the protein. So hydrophobics want to be on the center of the protein. If they're not, then they're being exposed to water, and this is somewhat unlikely. So what we do is we show it that little yellow, that yellow oily blob appears on hydrophobics that are exposed to the outside. And lastly, we have hydrogen bonds, which are actually a good indicator. If you have hydrogen bonds, it's usually means that your protein has something going for it, and they're really what holds the protein together in a lot of instances. So all of these visualizations are done, again, to keep things as accessible as possible to beginners. And part of the way that we do that is we hide a lot of information by default and let people sort of grow into the information and let them turn things on and off. And again, by default, most things are off, and people will turn them on when they need some specific piece of information. We also tried to keep the game sort of cartoonish looking and something that you might see in another game. So that, again, people aren't going to be totally confused when they come into this, and it won't just look like a piece of biochemistry software. So one area within visualizations, I was talking earlier about how Foldit is very iterative in its design, and we do a lot of refinement. Visualizations are one area where this has happened. There are 6 different screenshots here, each from a different stage of Foldit's development. And starting in the upper left and ending in the lower right, and hopefully those look quite a bit different to you. So one area in particular where we've done refinement, yeah?
Is it okay to ask questions?
Yeah, that's fine.
How do you know when the structure is correct?
So that's a good question. In general, we try and, we send out the puzzle and we look at the top solutions. So the real answer is we don't actually know a lot of the time, but in general, the higher the score in the game, the more correct the solution is, and the more likely it is to be closer to the actual native. And in some instances, as I'm going to show a little bit later, sometimes you get close enough that you can actually verify that the structure is correct. So you get your score to the point where you can actually take that, look at some experimental data, and decide that that's the correct solution.
And you stop iterating on it once scores kind of plateau?
Yeah, roughly. Sometimes we'll adjust puzzle parameters to try and deal with faults in the puzzle. But yeah, in general, they'll tend to plateau. So one area of refinement that we've done with visualizations is in hydrophobics. So if you remember, I was talking about hydrophobics, and they are represented, when they're exposed to the outside, that's indicated by this yellow blob that appears. It actually started out with this visualization, and there are several of them here that are actually exposed to the outside. They have this very slight orange glow to them, and it's really fuzzy and not very clear. So what we ended up doing is actually making them this discrete ball that appears, this discrete blob that appears, to make it much more clear that this is something that should be fixed. Oops. So again, on the note of trying to keep this accessible, like any good game, we have introductory levels. So you can actually see an introductory level being played here. In this level in particular, the person is learning to rotate the screen so that they can see the protein from a different angle. And they're also learning clashing. So they have to move these side chains apart in order to eliminate the clashes and pass the level. And when it finishes, you see some nice animations play and stars and fireworks. So we have all these people playing this game, but what have we actually learned so far? Well, the first thing is that our players are quite diverse across a bunch of different categories, gender, occupation, and country. We have a lot of international players, and we have people from all sorts of backgrounds. And one really interesting fact is where our players come from as far as biochemistry and biology go. So you can see that actually 3/4 of our players have one undergraduate course or less in biochemistry or biology. So with that in mind, how well are they actually doing? So again, we're doing protein structure prediction where they're trying to find the three-dimensional structure. So what we did is we took 10 different models, 10 different proteins that had actually been solved by some other lab somewhere, but had not been published. So we didn't know the answer, and the players did not know the answer. And we gave those to the players as puzzles, and we also sent them off to our computational methods to see how those would do on them as well. And what we found that the players actually can solve the structures. They can fix incorrectly folded proteins. And in particular, one area where they're very good within protein folding is moving through worse scores to get to better scores. So I'll show you an example of that in a second. And they actually managed to outperform computational methods in 5 of the 10 targets. So here's one example of where humans are able to look further away and go through worse scores to find a better solution. So this model right here that you see on the left is the protein that they started out with. We usually give players a starting model, and then they work on trying to manipulate that into what they think it should actually look like. So keep this model in mind. In particular, look at the blue tail that's been highlighted, and just sort of watch what this person does with that tail. Underneath the model, you'll also see a dot, which represents that model's score. So, and the time represents which solution this is in the series. So what the player does is they actually sort of start screwing up the protein and make it a little bit worse. And then they make it really bad, and it's sort of just falling apart. But then they sort of start piecing it back together. And in the end, it's actually higher than when they started. And if you kept your eye on the blue tail, you'll see before it was sort of sticking on the outside of the protein. Now it's been moved onto the inside of the protein, and these little arrows show up. Now what these arrows represent are something that we call a strand, which is a substructure within proteins. And what they've done is what we call a strand swap. So the turquoise arrow and the blue arrow were actually switched in the starting structure, and they've swapped these two things. And a result of that is that they've managed to create this sheet on the inside, which is when multiple strands come together. And doing this has actually resulted in a much better model. But a computer would not have been able to do this because it could not have pulled the protein far enough apart and seen that there was a better solution further away. So this is just a graph showing all of the solutions from that example that we just saw. So each dot on this graph represents a solution. And in general, the better solutions are the ones that are further down and more to the left. The ones that are further to the left mean that they're closer to the correct answer, and the ones that are further down just mean better energy. So you'll actually see most of the player solutions, or a big chunk of player solutions, is all by itself over in this corner. These solutions directly correspond to the solutions where people performed that strand swap that I was talking about, that player just demonstrated in the previous slide. And here you can actually see the path that player took. Each one of these dots is that player's steps through the protein as they folded it. So they managed to solve some structures from a set of already solved structures. So that's good for verifying that our technique actually does work. But we thought, well, why not apply them onto a real-world problem then? So with that, we took the Mason-Pfizer monkey virus retroviral protease, which is a protein related to AIDS in monkeys. Experimentalists had been working on this protein for 15 years, and computational methods had failed to solve this protein. We gave it to players for 3 weeks, and I think within 10 days, they had solved it. So an interesting fact about how they had solved it is it was actually the work of 3 different players who handed it off to one another in series to come up with the resulting solution, which was correct. And so what you can see in this slide is actually, it started on the left with the red line. That red line represents the structure as we gave it to them. And the first player took the red line and turned it into the yellow line. Now, the blue structure that you see, the blue lines that you see in each of these, represents the native fold of the protein. This is the correct answer. So the red one is actually fairly far off from it. But the first player took the red, turned it into the yellow. The second player took the yellow and turned it into the purple. And the last player took the purple and turned it into the green. And you might be able to tell that the green is actually much closer to the blue, the correct answer, than the starting structure, which is the red one. So by doing this, the players had actually gotten the model close enough that the experimentalists who had been working on this protein were able to take that model and combine it with their experimental data and determine that it was correct and resolve for the correct model. So we've shown that we can solve for the 3-dimensional structure.
So I'm going to ask the opposite of the question I asked you before. What do you give them to start with?
Usually what we give them to start with is some sort of 3-dimensional, it comes off of our servers. So
Just give them a random shape to start with?
no, it's actually, so we run our automated techniques on it, the computational techniques, and give them something that's like not completely terrible, but is like reasonable. And then from there, they'll actually refine it to the point where it's the correct answer.
You give them somewhat of an educated
Right.
guess.
It's like an educated guess, but it's probably wrong. We usually know it's wrong. So we've already shown that we can solve for the 3-dimensional structure, but we're actually really interested in how they do that, because this is how we can actually make Foldit scale and handle all sorts of problems. Because if we can figure out how they did that, that's really valuable to us. So this section is called algorithm discovery, and it focuses on how they actually fold. So the players have actually made their own website, this wiki, which documents all of their techniques that they've used while folding proteins. And it's a very good resource, and people are always directed to it as they join the game. But that wasn't really enough for us because it wasn't very concrete. So what we did is we created a thing called the cookbook within the game. And what this does is it lets players automate their strategies and sort of place them in series so that they can, instead of having to do the same repetitive work that they've been doing, they can set up the computer to do it for a particular strategy. And we call each of these strategies recipes within the game. And players can share them, and there's actually quite a lot of sharing that goes on. And there's some really good script writers out there who write these things. And initially, it was just through this GUI interface that you see here. The players came to us and said, hey, we want to be able to just like actually write code because we understand how to code. So we made a coding interface and implemented Lua in the game. But even that wasn't enough for them. So we actually hired one of the top script writers that was playing the game as a player and had him write code for us to make the scripting better. And that actually turned out to be a pretty cool thing, and it worked out well. So this is a graph showing different recipes. Each recipe is a column. Don't pay any attention to the colors, they're kind of meaningless. But the height of the column represents how much all players have used it. And in particular, you'll see there are two columns which are much higher than the others, and one that is very, very high. So when we were doing our analysis, we obviously wanted to look at this really often used recipe. And that algorithm was called Blue Fuse. And when we took a look at it, it actually looked really familiar to a lot of the biochemists. And so there's a reason for that. There is an algorithm that the Baker Lab is the biochemistry lab at the University of Washington, who we coordinate with on Foldit. The Baker Lab had developed this algorithm called Fast Relax. Now this algorithm was not published, and it's quite a bit better than any published algorithm. And so what the players had actually done is they had independently discovered this algorithm while playing a video game. And again, both of these algorithms are much better than what had actually been published. So the players had managed to find an algorithm that is as good as what scientists had developed in a lab. So we covered a little bit about how they've solved the three-dimensional structures and how they've actually done that with their different techniques. And the last big region is in protein design, which is actually, we have a three-dimensional structure that we want, and we want to know the sequence that actually results in that three-dimensional structure. And this is really important because, again, the three-dimensional structure determines the function of the protein. So if we can write a sequence that will make a particular shape, we can then write a sequence that will make a particular function of a protein. So this is the design interface within Foldit. And basically what it lets you do is insert amino acids into the sequence, and you can change existing amino acids to other amino acids. And protein design is also just a really cool area because it has the most direct ability to impact things. So players can make disease inhibitors, new biomaterials, and vaccines, and things like this. And it's also really cool because there's fairly direct interaction with the lab that we work with, the Baker Lab. So as players make their designs to, let's say, make a flu inhibitor, we actually take their top solutions and send them off to the lab to test and try and synthesize these proteins that they're coming up with. And so based on the results from those, we'll give them feedback as to how the tests went and what we expect are the issues with the current proteins that they've developed. And then we'll send off the puzzle again and give it to them again, and they'll have to make changes, and then we'll test it again. And there's a feedback loop going here in order to try and come up with the correct result. So in this area, the first Nature paper that we had was the 10 structures where the players had found 5 of the 10 better than the computers had. The second one was the monkey virus, where they had solved the structure that experimentals could not. And I think there was actually, the algorithm discovery might have been another Nature paper. I can't actually remember how many we've had so far. But this was actually the 4th one that we've had. And this protein that you see right here, as it started out, is a protein related to the Diels-Alder reaction, which is a very important reaction. And the discovery of this protein won the Nobel Prize in 1950. So what we were interested in is actually improving this reaction by modifying this protein. So this red structure that you see here is the protein as it started out. And one of the objectives of this protein is to sort of hold that turquoise piece in place. And so it does a reasonable job at that, but we decided to give it to the players and see if they can make it better. So what they came up with is this blue structure. Now, the main difference that you'll see between these is there's two helices. Helices are a substructure within proteins. And what they've done is they've constructed those up near the top of the protein. And these two helices sort of fold down on top of the turquoise part to hold it in place. And by doing this, they actually managed to increase the activity of the reaction by over 20 times. So again, Foldit players were able to do something that the people in the lab could not. So what's next? One area where we're interested in expanding into is providing some experimental data to the players. Right now, again, they're just given a starting structure and a score function and told, go find the highest score. And that's not actually the best situation, because we actually have experimental data for a lot of these proteins where it can help them find the correct structure. So one area where we have that is we have these incomplete electron densities. They come out of a particular method for trying to experimentally determine the protein, the structure of the protein. And we give them these densities to the players as a visualization, and they also impact the player's score. And their objective is to try and fit the protein into the gray blobby region. And so this is something that we're trying out and are excited about. The other really cool area that it's sort of expanded into is tools for scientists. So Foldit is a game. It's designed to be very easy to understand. You can tell a lot of things fairly easily. The interface is very nice. And those are actually very desirable traits of any sort of software. So the scientists in the Baker Lab actually are using this for their own protein design. And another area that we've expanded into is trying to make the interface with the game better. And one way that we're doing that is with the Connect. So we're setting it up so that people can actually manipulate with their hands the parts of the protein and push and pull them apart to try and get the structure correctly. Because when you think again about it, it's really a three-dimensional puzzle, and you feel like you should be able to just grab pieces and move them around. And another area is in education. So Foldit has already been used for education by many people. In high school classes, teachers will often set up a competition for their students, and their students will compete and try and solve a protein. And we're interested in facilitating this more. And so the way that we're doing that is we're actually going to set up an educational version of Foldit, which will try and teach basic ideas that are related to biochemistry in all of high school biology. So we're trying to really use this software to demonstrate some of the core principles of biochemistry that as it plays its role in biology. And another cool area is nanotechnology. This is now getting into stuff that is not related to Foldit, but is sort of inspired by the same sort of setup that Foldit has. So this is roughly what we call the DNA game. It doesn't really have a name at this point, but it obviously looks a little bit more kiddish at the moment. So this particular game actually involves making little machines out of DNA. And it turns out you can do some pretty interesting things with DNA. For example, you can make logic gates out of DNA. You can also make a sort of machine that will walk on other pieces of DNA and transport something. And you can also make DNA that will form massive boxes that you can store stuff inside of. And so the way that we're planning to use this is set it up as a sandbox inside of a Flash game online. And people will be taught the basic rules of how this particular exploit of DNA works, and they'll be able to use this to create all sorts of things in sort of a sandbox environment. And we'll get back their solutions and see what they're doing. And this has the potential to do a lot of really cool things at the cellular level. And so Foldit has shown us that you really can integrate humans and computers together to actually accomplish something that neither of them could alone. And games are a great interface for doing this. And we've only really scratched the surface of the different fields where this actually could be applied. And I hope that you guys will actually sort of think about this problem or different problems in this way and try and come up with similar techniques. And with that, thank you.
Something I was curious about is for those techniques for solving for all the folding, are you using search algorithms, or are you using like genetic algorithms or global optimization techniques?
So the way that the game actually uses a lot of these tools, but is directed by the players, the way that it works is we have an algorithm, like it's called a minimizer, and it effectively just like computes derivatives of the score function for—
So it's a gradient descent algorithm.
Yeah, gradient descent algorithm, exactly.
Okay.
So that's basically how it works.
So can you use the heuristics that you can learn by observing the players and then reincorporate those into your computational models to get the extra smarts that the humans have, reincorporate that? Have you gotten to that point yet?
Right, so that's sort of what we were attempting to accomplish with the algorithm discovery portion.
Okay.
So again, we initially started off just sort of like trying to watch what they were doing, and it turns out that's extremely hard because it's like, well, they're dragging this piece and then they're dragging that piece, and we don't really know what's going on. So our attempt with the cookbook that I showed is to actually help the players formalize and make concrete their strategies so that we can then take those and plug them back into the game, or back into the software that we were already using to predict proteins.
But it looks like the top one that the players discovered
was actually already implemented, but they didn't know.
Have there been any like new ones?
We haven't found any yet,
Okay.
but part of that, our hypothesis is that we haven't given them the correct tools to be able to do a lot of these things, because the tools that are actually given to the players are a subset of all of the tools that are available within the biochemistry software that we're sort of based on. And if we give them more hooks into this code and allow them to mess with more of the parameters, we think we'll get more algorithms that have not been discovered and that will actually make a big impact into how proteins are folded.
Do you have a legal disclaimer for the players on this, that they do not get monetary compensation or
Actually,
academic credit or anything like that?
So this has sort of been a recent problem for us. And well, we're sort of preempting the problem. We're trying to come up with a sort of license. But really, I think the conclusion that we've come to is that this is actually determined already by copyright law. And so we don't really have too much control in the matter. Basically, if some result does come out of this and there's money involved, the players actually have a stake in it. So they do have the ability to be compensated. But yeah, we're still working on that part, in short.
Yeah, when you do a computation like this, you have a local minimum, global minimum. Okay, so you kind of lock into the system. When you introduce a human intelligence into the system, you break and make it. Okay, so that is not available in a computational mode.
Right.
So that is what makes a significant difference in this folder.
I think so, yes.
I'm wondering, what's the largest size of the proteins that computer can simulate? I mean, the largest by the computer power, the limitation of the computer power. The other is by the degrees of freedom. And I'm wondering that when the size exceeds a certain size, then degrees of freedom is so large that it will be a chaotic system that you probably will not be able to model.
Yeah. So that definitely is a big problem, particularly so in the game, because we have to display all this stuff. So it actually scales worse in a lot of ways than the computational methods, which don't have to display this and which don't have the overhead that the game has. So the computational methods, I think, can handle on the order up to like 1,000 or 2,000 residues, 2,000 amino acids, somewhere around that range. I don't actually know because I don't actually use that software that much. Foldit itself actually can only handle about 200. But the important part is not so much the models that players are working on directly. It's more what we're interested in getting out of it is algorithms, again, because we really want to abstract the principles that they're using to fold proteins and apply those back onto the computational models. And the other thing that we're really interested in is protein design. And design proteins don't actually need to be that big in order to do interesting things. So that's sort of where we've been focusing our efforts recently. Does that answer your question?
Why is there such a focus on, ultimately, like you just said, getting the algorithms back onto the computational side? If you made a point several times of saying that computations are still going to fall, computational capabilities are going to fall short. So I would think that's true, even as your algorithms get better. The more complex the protein, you're still going to fall short of the computer.
Yeah, it's a completely reasonable concern. I mean, the problem is that the game itself does not actually scale that well. We only have so many players. They can only work on so many proteins, and we actually can't solve anything, or they have trouble with dealing with things over 200 amino acids long.
So players themselves can't really handle something
that sort of remains to be seen. We're planning on sort of developing techniques which will let players work on sub-problems within a particular protein to be able to tackle larger things.
Okay.
But as it stands right now, they have trouble, and the software has trouble with things over 200 amino acids.
Okay.
Have other disciplines approached your team to try to apply this thing? I know you mentioned a little bit with like nanotechnology type stuff. Are people coming to you and your team is helping them build out the infrastructure, or do you have any thoughts of using opening this up as like a cloud platform so people submit to you your problem, you help build out the interface, and then?
I mean, that's actually fairly a good description of sort of what our lab does. We're trying to work in a lot of different directions on solving different problems with this sort of technique. But other people have actually been working on things like this as well. Adrian, I don't actually know how to pronounce his name truly.
I'm not sure.
Okay, but anyway, he actually helped co-create Foldit. And from there, he actually moved to a different university and is working on a project called Eterna, which is actually an RNA folding game, which is a different domain, very related, but it's using the same technique to try and solve real-world problems.
I noticed in one of your slides that there's a disproportionate, it was more than 75% of the players were men rather than women.
Mm-hmm.
So my question, and I know this is kind of outside the domain or the responsibility of Foldit to do this, but given that disproportionate or the relatively few number of women, can you use that as a catalyst to be more proactive, to get more women into the STEM fields by taking kind of a game-based approach as opposed to kind of a more traditional approach?
Yeah, so the answer is you probably could. We haven't actually done too much in terms of that. Usually where we get most of our players is we'll have some paper that is published, particularly with the monkey virus paper. You may have heard about it in the news, and it was pretty much everywhere. And we had several thousand people coming into the game and playing our game at the same time, which actually crashed the servers and stuff. Yeah, so we haven't really made any steps towards trying to do that. We have a lot of things on our hands anyway to begin
I wouldn't
with.
have expected you to.
Yeah.
I just was curious to know if that's ever been discussed. Of how
Yeah, we haven't really discussed that too much.
Okay.
How do you market the game? Or how do you spread word of this?
Again, like I said, part of it is coming to talks like this and people check it out, and the other is publishing papers. And then after people see this article on a protein folding video game, they're curious and obviously want to try and check it out and see what everything's about. So that basically is how we've handled these things.
So do biochemists do better, or
What was that?
Have you looked at?
Oh no, that's actually something that I neglected to mention. So it actually turns out a lot of our top players are not biochemists, which is really interesting because these are people that are totally untrained, and it's only through the game that they've learned these techniques, and everything has been abstracted away.
So the learning curve for the biochemist is lower, but
Probably a little bit lower. I mean, they'll know what a side chain is. They'll know what the backbone of a protein is. They'll have some basic knowledge. Yeah.
Yeah.
So you mentioned there was a client that gets installed on the person's computer.
Mhm.
At this point, this is like Windows PC only, is that right?
No, it's actually available on Mac, Windows PC, and Linux. But it's not available on mobile platforms. We get a lot of requests for that. But the problem is just that mobile platforms, I don't think, have the power to be able to actually do something like protein folding. So even though the humans are helping the computers in this case, it still takes a lot of computational power because you're still doing like a gradient descent minimization of energy. And that really requires your computer to crank away really hard and uses a lot of processing power in order to do that.
So the client side is actually doing a lot of the CPU?
Yes.
Okay.
It's very CPU intensive.
And then are you must you be continuously connected to your system via the internet to play the game, or can you download a model and then play with it and then upload the results later? Is that
You
possible?
can. The way that people usually do is they will sign into the game and then just as they're doing that, it'll send us a solution like every 3 to 5 minutes, I think, of indicating their progress. But you can play offline as well, but it won't really integrate with the scoreboard at that point, which is what drives a lot of people to play our game. So most people just play online. They'll get disconnected sometimes, but it's really not a big issue because you just reload the game and then it'll upload your solution. Yeah.
I think you touched on this. Is there a project underway that is now using Foldit on the front end and then going to the lab? You talked about protein design. Is there a significant one ongoing where the scientists have said, gee, let's use the game first because I'm interested in a particular protein or region or something like that, and then go back to the lab?
So you're saying the scientists using the game directly?
To use it as a design element, you said protein design, before going to the lab. In other words, try out your ideas using the game.
Yeah, again, I said scientists, the people in the Baker Lab actually are using the software to do their initial designs before going back to lab work. So yes, scientists are using this as sort of biochemistry software because it actually turns out to be very convenient to do protein design within Foldit, and there really isn't software that sort of captures the functionality that Foldit has.
Have there been other groups now pick up Foldit other than University of Washington?
So our servers are set up to be like kind of proprietary, and the code base itself is also proprietary. So it's not really open at the moment. We're interested in trying to make some of it open source, particularly because it'll help us fix bugs because the players themselves can do it.
Fix it.
Yeah, but so far it's just us.
Okay.
Yeah.
So what's like the development time on something like this? So if another group or another industry wanted to develop a game, like how long? I mean, obviously it's all dependent on the complexity of what you're trying to solve, but in terms of Foldit, how long did it take to develop to get to this point?
So it's been in development, I think, over 3 years. And so that's for this point. In order to just get the game initially released, I don't think it actually took that long. Of course, we have some very smart people working on it in the back end. The guy that I work under, Seth Cooper, just got his PhD and on Foldit. And yeah, he's unreasonably smart and can code things up like this very well. But yeah, it didn't take that long to my knowledge.
You mentioned there was a group play mode. Can you talk more about how that works?
Yeah, so it's just like the solo play. You'll start out with a protein, and at some point when you think you have something worth sharing, you can share that solution to your group. And your group members can see that you've shared it, and they can load it into their game client. And when that actually happens, it'll be considered like a group solution, and they'll also get an evolver score, which an evolver score basically means you're using someone else's solution, and you'll go on an evolver scoreboard, and that solution will also show up on your group scoreboard. I don't know if that was clear or not, but
Yeah, it makes sense. So essentially, the puzzle is the same. The solution would be the same. It's just that your rank is according to, oh, well, more than one person worked on this, so let's factor that
Exactly.
into ranking.
Right.
And the rank is for the length of time that you publish that puzzle, is that right?
Right, so when the puzzle closes, we have some additional elements that encapsulate all of the puzzles. And this is like the global leaderboards of the game. So when a puzzle closes, usually they're worth about 100 points. And based on your rank, you'll get some number of points, and those points will be added to your total. And so this total is like all of your puzzle results in the past 4 months. Is the way that it's set up currently. And so some players will have like 4,500 points or something, and other players will be lower depending on how they performed in puzzles over that last period.
Do people have the capability to publish their solutions, not to any particular small group, like their friends, but to like the general community? My thought is if you could hit like kind of more iterations on the same puzzle, you might be able to find a more optimized solution if it's more widely shared.
So we've actually tried that in a mode called All Hands, which was basically every solution people can share and amongst a global group where everyone's just a part of it. And what we found is this actually kind of hurts the game, not helps it, which is somewhat counterintuitive because you expect these people are collaborating. But what actually happens is everybody starts working on this same sort of solution. And everybody is they see a good solution and they'll just work on trying to improve that one. And what ends up happening is people just drill straight down, like in the energy function, don't change the structure very much. They just try and focus on the score and aren't willing to make changes. So yeah, we've basically tried that and come to the conclusion that it's actually somewhat harmful to how people fold proteins. But we're also interested in different ways of doing that perhaps will still allow for the same level of collaboration, but not hurt the actual results.
The interface looks very easy to use, but when it comes down to it, you still have lay people manipulating amino acids and whatnot. Has there been any discussion about sticking on another layer of abstraction? And so you're not, in the sense that you're not manipulating a molecule, but manipulating, you're designing like a roller coaster that models back to.
Yeah, I mean, it all depends on what you sort of select as what you think is the correct layer of abstraction. And this just happened to be what we did for Foldit, and it's worked out fairly well. But I agree, you could probably reduce this problem of protein folding into a higher, more abstract level, and potentially that could work as well. But really the reason I think usually for doing that is that people are better at solving a problem when it fits in, is simpler, and they can understand it in the frame of that problem. So I think people are capable of understanding protein folding as it's shown in Foldit. So I'm not sure if making it more abstract would help, but that's an open problem. I don't actually know the answer.
Yeah, I just wonder about those people that don't care about protein folding. If you make it more of a game, that you get when people are actually using it, even if they don't realize what it is they're actually doing.
Is there any kind of reward for your top scores?
So there are like achievements in the game, and people also like just being on top of the scoreboard. People really like that. So it's a big motivating force for a lot of people. We're planning on adding some additional things like an achievement for reaching the top of the scoreboard, something that's really like will stick and that you can show off.
Like a trophy of some kind.
Yeah, and of course, there are also the benefits of being listed on a scientific publication, as some of our players have actually done. So actually, I think it was the monkey virus paper that I was talking about. We asked the players if they wanted to be listed as authors on the paper. And they actually declined and said they just want to have their group listed because it was really a group effort. So if you look on the paper that was related to solving the structure, it'll have the Foldit Contenders group, because the Contenders was the group that solved the structure.
Is the environment more like, is it more single players, or are there more teams of groups that tackle the problem?
I'd say it's pretty competitive in both respects. It's really both. There's a lot of group play, there's a lot of competition between the groups. There's one group in particular that is decently ahead of the other groups, but there's still lots of other groups that are competing and doing very well.
Sort of like the top scorers all from the teams as opposed to individuals.
Usually I think that is the case, but not always. Sometimes a soloist who is not part of a group will come up with a solution that is better than any of the group's solutions.
Have you come across any savants, any one person who just...
We've had several.
Yeah.
Yeah, we've actually flown in to Seattle, some of them, and sort of just talked with them to sort of figure out what they're doing. And David Baker, who's in charge of the Baker Lab at UW, who does the biochemistry side of this, has met with some of them, and he's just like, sort of like looks at them strangely, like they're performing some sort of black magic on the protein.
I was just curious, I saw one of the top scores at HS after we go to high school.
What was that?
Meeting. One of the multiplayer ones said HS after it. Is that a high school that's playing?
Are you talking about the...
It was in the rankings, I saw it.
Yeah,
Oh,
the ranking.
I'm not actually sure. I don't think there are any high school teams actively playing, but
It looked like it was like a city and then it said HS.
it could be. I don't actually know. We get a lot of groups that will come for a few weeks and then stop playing puzzles and things like that. But usually when it's an education-based thing, a teacher in, say, high school will set up a competition. So we have an interface for allowing players to create a competition just among their group, like be it their high school or whatever. And they can have just the people that are part of that compete on a puzzle that they set up. So that's usually what happens when people set up something like this for their class. Good.
Great.
Thank you.
So before y'all take off, I just wanted to thank Jeff again for coming. I think this just continues to fit very nicely with this idea of exploring new ideas and ways of problem solving. So thanks very much for coming. And a small token of our appreciation for visiting the center is a montage from Spaceport. We can ship it to you if it won't fit in the overhead.
Yeah, it probably won't.
Thanks again for coming.
Thank you.
And I have to imagine that was by far the single most number of questions we've gotten. So obviously a great talk. So thanks again.
Thank you.
Thank everyone for joining online.
Yeah, I probably have to do that.